now I'm recording. Let me go back. Okay. Again, that lab notebook check. So that would be genetic engineering and then dental caries, which I'll talk to you about super briefly here in just a second. So those two things you should have completed, again, just like you would if you were in the classroom, just with the lab notebook entries, including the conclusion questions that are from the lab manual. So get those two things, you can just packet them together in however you want to, and I'll just have, there's one spot to drop all of that stuff. So if you want to upload 2 files, genetic engineering, dental caries, that's fine. Or if you want to combine them into one, that's fine too, but it'll all go into one Canvas drop. So those are, I just put some arbitrary points to that one. So it'll be every single week you'll upload, and I'll specify exactly the labs that you should be uploading every week. For example, down here under upcoming assignments, you see the second one here, lab notebook check. This is for next week. This will be the normal floor exercise, and then the lysozyme lab that we will talk about or start next week. So both of those will be compiled together, and you'll submit those in another Canvas drop by next Friday. Okay, so that'll be every week. Of course, you'll only have this week and next week, and then you have spring break. So when we come back from spring break, we'll pick back up again and just do it every week from then on. Questions on that lab notebook check? Hold on, let me pull up the chat thing just so I can see if anyone has a question. If anyone has a question, you can raise your hand and unmute yourself if you want to. Do we understand that lab notebook check? Yeah, no, okay. Okay, thank you. Up here also you have that the rest of these things are due on Monday by midnight. So you have parasitic protozoa and helminths. That's the assignment I provided for you last week. It's that PowerPoint that you're essentially just going through and filling with information you can glean out of your textbook and your photographic atlas. And you'll submit that one on Monday, and then on Tuesday we'll have about a 5 or 10 minute period where we can discuss that one in class or in our lecture series. After Thursday, today and Thursday, we're going through the rest of this basic stuff on genetics, which is replication, transcription, and translation. And hopefully by Thursday, we'll have touched on genetic mutations. So I've already posted your in-class activity for that one, which is going through a transcription translation problem with genetic mutations. And that'll be due on Monday. And then I'll give you an extra credit assignment, and I'll talk to you guys about that more on Thursday. With what you can do for this. It's called Say It With DNA, kind of a fun little activity. And then just so you're keeping your eyes up on upcoming assignments, this is the stuff that's due within now to the next week. And then after that, you have a few more assignments. This normal floor exercise that you're starting today, you have until next Friday to complete the lab notebook part of it, but next Wednesday, I want you guys to submit the worksheet for it. So there's 2 separate due dates for that one. You have that lecture exam. I'm going to open it. Hold on just a second, Jeff. I'm going to open that lecture exam for you after our discussion time on Thursday. So we'll actually do next week's Tuesday and Thursday regular discussion time here. And then as soon as this meeting is over is when I'll have that exam open for you, and it'll be open until Monday the 13th on at midnight. And so you'll just have to choose a 2-hour interval sometime within there to take that exam. And no, don't ask me about that yet. I haven't finished. I haven't gotten around to that one yet. You do have a few other things. There's a couple of your extra credit things, that vocabulary list. I have to make that due right at that start of the exam opening. So if you wanted to get credit for that one, that'll be due by next Thursday by the time we hit this lecture discussion time. And then you do have that Contagion movie extra credit. I want to mention this one, that Contagion movie. I just watched it again recently with my family. It is really applicable. And by the way, I just found out that the director, one of the directors of that movie is actually an epidemiologist who was involved in the smallpox epidemic way back in the day. And he helped eradicate this from the globe back in the 70s. So it is so applicable to what we're facing today because it's based on scientific, like demonstrated scientific knowledge on how epidemics spread in a population. So I definitely recommend watching that. And I will choose one day, probably Friday, probably Friday, April 3rd. I could choose a different day, but that I'll actually live stream that movie for you. If you want to get that extra credit and you don't want to purchase that movie yourself or don't have another way to watch it, I'll just do a Zoom conferencing. I'll share my screen. You can just watch the whole thing. So maybe I'll ask you guys when a good date to do that one is so you can watch it if you choose to. Okay, let me answer some of your questions. Jeff asked, oh, Oh, about the protozoa and Hellman's PowerPoint. Yeah, I said that you could do this directly on the PowerPoint slides, or you can do it separately on a sheet of paper, on a, by hand, or on a Word doc or something like that, where you're just filling in all of the detailed information and then upload that. Totally fine. Either way will work just fine. Yeah. And then Katya asked about the vocab lists. Vocab list 1, I don't have a deadline for, but I just mentioned vocab list 2 will be due on the date of the time that I open up that exam. So down here on Thursday, April 2nd. There's some other things coming up. And then I did finally put a deadline for your first part of that unknown exercise, because I do need to keep you guys on track. This is actually way farther out than you should have it done, but this is that dichotomous key and biochemical results chart. At the very latest, I'm going to make it due on April 17th, but again, that's just to make sure that you guys are staying on track. You really should have this done sooner than that. I would urge you to work on that over the spring break. It's like popcorn in my ears. Work on that over spring break. Don't rush it, but definitely when you have that break, quote unquote, if you actually have that from your classes over spring break, then try to get that dichotomous key biochemical results chart and dichotomous key completed. Don't forget, you guys are totally welcome to work with each other. If you've actually downloaded the Confer Zoom thing, like if you have made your own accounts and even just one person from your group has made their account, then you can have your own meetings through Zoom. CRC has, I know the instructors have our accounts through here. I don't know if students can make this, but even Zoom conferencing, not Confer Zoom, but Zoom is free, and it's free to have an account with them. And once you have an account, it's free to host meetings up to 45 minutes long, and then they don't really penalize you for longer than that if you kind of go over. So if you guys want to have this as your medium, it's really easy to use as a host. So I really, really recommend you work together with people and use Zoom if you want to. And like you saw, you can share whiteboards and write things out and share screens and everything. Otherwise, you have your own means of communication with each other, but that would be a really smart thing. So let me check with you guys. You also, I didn't post on here yet, but you do have those presentations coming up pretty soon. I will probably tomorrow, this evening or tomorrow, since I'm now caught up with some of this stuff, I will go in and make those groups I told you I would do. So I'm going to start by making your groups according to your upcoming presentations, so that on Canvas itself, you'll physically have groups on there that you can click on and then exchange information that way. You can share drives, a Google Drive, where you do Google Docs and make your PowerPoint presentation that way if you want to. And we will do, it seems like this is where we're going to be for a while, so we will do our presentations through this medium here. And so you guys do still have to have those instructor meetings with me. And so what I'm going to do is during your instructor meeting, which is just going to be anytime you schedule a Zoom conference with me, you and your group, I'm going to have you all practice doing a super short presentation where you essentially just pass the baton back and forth, and each person will have the opportunity to say something. And that way, at least you've had the experience of practicing with an audience of each person presenting information for a presentation. And that's how we'll do these ones where your group will just unmute themselves sequentially, and then you can each speak their presentation this way. If there's any conflict, of course, you can do a pre-recording, but ideally we would do it live here on this thing, and then we'll have a little discussion part after each presentation, which also then brings me to a question I want to ask you. Let me see if I can make a poll for you. So I would like for us to have a little bit more time on here that we can devote to laboratory discussions, because I've been posting a bunch of stuff for labs and some overviews and etc., but we don't have any time that we actually interface where you can ask me questions and I can clarify things for lab, and I don't want to eat it into our lecture time. So what I'd like for us to do, and I'm hesitant infringing upon more of your computer time, but I know that we're physically scheduled to be in lab for 2 hours each Tuesday and Thursday. So I want to see if we can, like, ideally, I would love for us to find a time either 20 minutes before this, like basically from 5 to 5:30, or from 7 to 7:30 on Tuesday and Thursday, that we could just devote to laboratory time. It'd be great if I could have all 35 of you guys at the same time, so I don't have to split it apart. But if that doesn't work and I need to split it in half and do half of you guys before and half after, that would be okay. Let me see if I can just add a quick question here and see what you guys think about when a good time to meet on Zoom would be, either before or after. Let's see. Okay, I'm having some challenges here. What I think I'm gonna do is, since my poll question is not letting me edit it right now. Add a question. No, okay, I can't add a question on here right now. And I really need everyone's input. So if I can't figure out this poll question and add it while we're talking here today, then I will submit to you guys all a, what is it called? A doodle, whatever that doodle thing is, scheduler thing. I forgot what it's called. Anyway, I'll send that out to the whole class, and then you guys can respond to the most optimal time. Maybe give me a couple of different time periods that work for, well, the most optimal time for everyone for a lab discussion time. And then we'll get that implemented, hopefully starting on Thursday, that every Tuesday and Thursday we'll have that just short discussion time for the lab-based exercises. Okay, straw poll. Okay, thanks, Jordan. I think that's the majority of the stuff that I wanted to get on for the logistics of everything that's here. Okay, don't worry about commenting on here. I'll just send out a poll because I just really want to get the best times. But I will post an announcement. Actually, I'll just email everyone. I'll email the course by tomorrow once I get an idea as to what that lab time will be. And so we'll just have that Zoom conferencing. And again, it might just be during your regular lab time, but just for about a half-hour window of that time period so that we can go over some of these things. Let's run over to this dental caries exercise, just because we don't have that scheduled that time. So I do want to make sure you guys are updated. Oh, I see. Thanks, Jenny. That's a good idea. Okay, let me do this first, and I'll get you guys to, as soon as I get you guys to a place where I can break you out into a discussion group, then I'll go do that poll thing while you guys are working on something. So let's finish this stuff up first. I'm just taking you back over to our main page. I think I'm still sharing the screen with you. Down under the lab material, you already see this genetic engineering exercise. Everything is up for you there, including some really helpful videos. On Thursday, which is essentially the day before the stuff will be due, we'll have that lab time where we can discuss this a little bit more thoroughly, and I'll talk to you guys over that one at that point. But I just want to show you this dental caries lab and the normal flora exercise that I've just posted yesterday and today. This dental caries lab, this is a pretty straightforward one. You guys were asked to do this pre-lab that was due today. That's really just based on your own dental hygiene. And so I'm going to use that pre-lab and the thought questions that were in there for you guys to essentially figure out what you would have expected to see or make a prediction, based on your personal dental caries susceptibility, like your history of cavities and your oral hygiene. So I have here a cute little pregnant woman who's talking over the Snyder-Agar test, which is what we use in this lab. There's no visual, there's no evidence, or there's no tubes that she's showing you, but this is exactly what I would be talking to you guys about in the laboratory. So watch that. It's short, it's 5 minutes or something. Watch that short video, and then you can answer or complete your lab notebook based on these results here. These are actual results from our class, where one of these is a positive and one is a negative. Oh, I tell you already. So this is what a positive tube looks like. This is an uninoculated negative tube, A and B. And then my question down here for you is, make a prediction of when you would expect your Snyder's-Agar tube to turn yellow. If that doesn't make sense to you yet, that means you haven't watched this video yet or looked at the lab, which is fine. But at some point, take a look at that, read through the lab exercise, and look at the chart we have for you there, which is the same one that she talks about in the video, and indicate what, or write down essentially what your tube, what you would expect your results would have looked like, and then discuss those based on your understanding of this lab. So this is just kind of a predictive lab, but use these results here to say this is what my tube would have looked like at this point, and this is what that means, okay? There's no associated worksheet for this one. This is simply your lab notebook entry. Questions on the dental caries lab? Anyone? No? Okay. So one more thing, let's go back over to our modules. The other lab that I posted today, and I just finished the video for this one, so I'll upload it in just a minute. This is the normal flora exercise. So in lab, we would have had you swab 3 different areas of the body, your nasal cavity, your throat, and your rectum. And after streaking for isolation, then we would have you characterize different organisms based on their growth characteristics on selective and or differential media, and some other tests. So gram stain for the nasal cavity, and catalase test and gram stain for your throat. And then for the rectum, we would have you do the whole INVIC series. So these are biochemical tests that we use to differentiate coliform bacteria. And so I put together the results for like some expected results or really common results that you would see for each of these different body cavities or body areas. And your assignment is to essentially work through this results chart or this results PDF that I have for you. And you're just identifying two different organisms. And for nasal cavity, there's only 2 that I've shown you. For the throat, I've identified 3 different ones. And for the rectum, I've identified 3 of them. So you just have to choose 2 of them for each of these body sites. And maybe my internet connection is too slow to show you what this thing looks like. But once you look over all of those results, you will be, again, choosing 2 of them and then interpret the results. You need to, in your notebook, write down what the gram stain means. So I just have a picture for you. You identify all the basic characteristics that we're asking you for. Anytime you write down cellular morphology, indicate their growth characteristics on the media that we're using. And then with this chart that I put on here for the throat and the nose, you can identify genus and species of the organism that you've chosen. And then for the rectal one, this one is based on a chart that's in your actual lab manual itself. So for this one, you'll have to just choose one of the 3 organisms that I put on that chart or on that PDF and go through all of the characteristics based on what you see there. So the gram stain, MRVP, citrate, et cetera. Again, in your notebook, you're going to write down the results for all of those things and what that means. So whatever it is, you would have MR positive or something and tell me what that means. And then at the end of all of that stuff, then you come up with genus and species identification. So all of that goes in your notebook itself. In addition, there is the normal flora worksheet right here that I've made, just extra questions that you can answer if you pull that up, by the way. Most of those questions I answer in this video that I'm going to be uploading this as soon as we're done here. So you'll have most of those answers. I'm just going to minimize that. So you have most of those answers already that go on that worksheet. The rest of them are just some thought questions to add on to this one. So for the normal flora exercise, as mentioned, this worksheet itself has a due date of Wednesday of next week. And then the rest of this stuff, all of these details that goes into your lab notebook itself, then this notebook check will be the second one next week on Friday. All of this stuff is written down here, and it's written down in the normal flora results thing, and it's written down in your lab manual. So you have all of this information. I know I just said a bunch of stuff. I also am finally recording this lecture discussion time, so I'll post this one also under modules so you can listen to me again if you want to. Okay, so those are your 2 labs for this week that you're working through. Don't forget about your parasitic protozoan helminth and genetic engineering. You should be wrapping that one up. And again, we'll discuss that one in detail on Thursday in our lecture lab time. And then you have the, I think that's it. Oh, and then unknown exercises. Okay, I think that I've covered everything that I wanted to get to. Hopefully you saw I finished up your genetics, your first section of genetics. So all of the videos are up for transcription, translation, and replication. Tomorrow I'll finish the videos for the rest of that chapter and get them uploaded for you too. So you'll have everything you need. And then we'll, I'm going to give us all the way through till Thursday next week to finish our discussions for that one. So we'll get through everything before that first exam is, or before the next exam. Okay, questions on any of that stuff? Anything from what I've talked about, logistical things? Everyone's having an okay time, hopefully navigating everything and finding access to things. Yeah, okay. I don't even see most of you. Okay. Okay, so I'm going to stop this screen sharing and I'm going to switch over to our genetics. And what I'm going to do is just, I'm hoping, I know that I've been watching those YouTube views, and I see that not many of you have watched this stuff yet. So I know that you haven't seen this video and you haven't seen these slides yet, but I'm still not going to spend a ton of time walking through each of them. I think this is kind of boring and a waste of all of our time. But what I'd like to do is highlight the key steps in DNA replication and then break you guys out so that you can at least on your own try to replicate this thing or with your small groups, and also give you more practice with sharing whiteboards and stuff like that. Okay. So let me move things around a little bit so I have my screen available. Oh, yeah. Okay. Hold on. There we go. Okay. So last week in our short lecture discussion group, we talked through the structure of DNA, and I had you guys write out that double-stranded DNA molecule. I want you to refresh that in your brain and remember what that molecule looks like so you remind yourself of where that 3' carbon is, because it will become relevant as we discuss through this process of DNA replication. These concepts here, I don't really want to go over in detail, but there are 2 terms that are discussed in prokaryotic DNA replication. The first is bidirectional, and bidirectional references the fact that this is a circle. Instead of eukaryotic cells, we have linear DNA. In prokaryotic cells, they are circular, and therefore when that circle is replicated, it's opened, and as it peels itself apart, both of those templates are not only replicated, but replicated in both directions. So as the circle peels open, which is kind of what you see in your screen here, right? Imagine this and this that's it's pointing to are essentially, as you're peeling those open, it's where those 2 circles are still connected down there. And so bidirectional means that both of those directions, to the left and to the right, essentially, as you open up that circle, are being replicated at the same time. So a full replication event for prokaryotic cells is essentially when both of those 2 circles open all the way to the bottom, and then those 2 strands are essentially disconnected at the very bottom. It's hard to show you in picture format, it's hard to describe it, but if you're imagining this was once a double-stranded DNA molecule, it was open, and here is the opening part. Here's what we call a replication fork. There are 2 of them because this is bidirectional, so both sides of this circle are being replicated at the same time. And this continues until both of those circles essentially have been replicated all the way down to the bottom, and then those new strands are separated at the very end. So that's this term bidirectional. It just means 2 directions. The replication is happening in both directions. And then semi-conservative references the fact that during a replication event, you'll have, let's see, let me draw this in 2 different colors. So during a replication event, let's say here is our original double-stranded DNA molecule. And we know we have different orientations, right? Maybe this is 3 to 5 orientation, and our complement is 5 to 3. And during replication, these guys will separate, and both of them will be templates. And this term templates, you know, references like if you have a Xerox machine or a copy machine, the template is that piece of paper that's going to be copied, right? Same thing here. So both of these will be templates, which means both of them will be copied. So during a replication event or after replication event, that 5' strand will be a template, and a newly replicated strand will be made, I don't know why it does that sometimes, will be made complementary to that original strand, and this will be in the opposite orientation, 5 to 3. So blue would be the new strand, and red is the old strand. Over here on the other strand that is generated from this replication event, if this was the template for this new strand, then we have the outer template 5 to 3. The outer strand is our template here. So these guys are called templates. And this is also considered like the old strand, if you want to say it like that, right? The old strand. Then a newly synthesized strand will be made in the opposite or the complementary orientation. So this newly synthesized strand is going to be complementary, right? This is new. So this is called semi-conservative. Semi means part, and that this means that each of these new double-stranded DNA molecules has one old strand and one new strand. One old strand and one new strand. Now both of these guys should be exactly identical, right? They should be exactly identical because the old strand here is complementary to the old strand here. Therefore, these guys should be exactly the same as these guys over here. I will tell you though that DNA replication is inherently error prone, just to keep that in mind. There is always errors or mutations that are incorporated during a replication event. We'll talk about that more on Thursday and Tuesday of next week. But essentially these guys should be exactly the same, but semi-conservative, one old and one new. This is just showing you again the concept of bidirectional replication. And what we're going to do is focus on what's happening at one of these two sites of replication. And this is called a replication fork. Replication fork, that's the term. That's the site of replication. So we're going to enhance this view right here and just walk through the process of DNA replication so you can see what's happening at this replication fork. Okay, here we go. So at this replication fork, and again, there's lots more, probably 10 minutes more of conversation on the lecture videos. So go back over to there. Don't let this plant the lecture videos that I've pre-recorded already for you. But I'm just trying to get to this point here. So let's start here. Make this bigger. All right, we're going to draw DNA replication and we're going to do one replication fork. So let's say, hold on, let me move some things out of the way so I have some more space. Okay, we're doing DNA replication. So DNA replication begins at something called the origin of replication, or the ORI site. I'm going to just draw it in here and say that somewhere over here, and I'm drawing one replication fork here. Okay. So at one point, this double-stranded DNA molecule was still closed in a big circle. And at one particular spot on that chromosome called the ORI site, or the origin of replication, the ORI, or the origin of replication. This is where particular machinery recognize a specific complex or specific sequence of DNA and bind to that double-stranded DNA molecule and nick that molecule open. So the ORI is where the replication machinery will bind and begin this replication process. So here we have the exposure of all of these nucleotides. And if you recall, nucleotides really, they're very reactive, right? Those nitrogenous bases really don't like to be essentially unbound. So they will naturally just close right back up again. But there are key components to our replication process that prevents that. That's called re-annealing, that closing back up again from happening. Okay, so step one is essentially the opening of our double-stranded DNA molecule at the origin of replication. There are two major enzymes. Now remember, I'm simplifying this whole thing, but that's okay. So let's draw our first enzyme in blue. Nice thing about this, I don't ever run out of ink, right? So over here at the side, let's write our first key enzyme complex here, which is called the topoisomerase. One is oh my gosh, sorry. It's very cold out here. My hands are starting to freeze. Topoisomerase. Note these guys are all ACEs, so they're all enzymes. The topoisomerase. I'm going to put shorthand. These guys unwind the DNA. Unwind. Now, this is like if you were to take a rope, a strand of, a rope, and you were to disconnect or de-thread the strands of the rope and you start pulling it apart, or even a thread or something, or even your hair, you would note that as you pull those strands apart of that rope, then the coiled part just gets tighter and tighter and tighter. So the job of the topoisomerase is essentially to remove the tension of the tightly coiled double-stranded DNA molecule, right? So this is why we call it unwinding. It's actually removing the tension of the coiling of that DNA, but you don't have to know that. Just consider it's just unwinding that strand so that replication can occur. This next enzyme, and both of these things occur kind of simultaneously, but we'll just keep them in this order. The next enzyme comes through essentially this unwound DNA, and it unzips it or breaks the hydrogen bonds. This is called the helicase. Again, an enzyme. All of these are enzyme complexes, but we're keeping it simple. And we're going to say this guy unzips that double-stranded DNA molecule at those nitrogenous bases. So this will break those bonds, those hydrogen bonds. Breaks hydrogen bonds. Whoops. Breaks hydrogen bonds between I need a bigger screen. Breaks hydrogen bonds between. So if they're breaking hydrogen bonds between these nitrogenous bases, would that be adjacent nucleotides or complementary nucleotides? Do you think the helicase, you can answer this one, does the helicase break hydrogen bonds between adjacent nucleotides or complementary nucleotides? Good, good. Yep, between complementary nucleotides. Complementary. Okay, so the bonds are broken, and again, these nucleotides really are very reactive, so they will, it's called re-anneal very, very quickly. But there are proteins that come and bind right away to prevent the re-annealing. These are called single-stranded binding proteins. And they bind on both of these guys. Okay, so this is number 3. Single-stranded binding proteins, or you can call them SSBP or SSBPs, single-stranded binding proteins. Their job is to prevent re-annealing of the double strand. Okay, so they essentially just keep those two strands open to prevent them from closing back up again. All right, now we can have our machinery bind to these things, and I'm going to show you on both of these strands. The process is exactly the same, but it's going to look a little bit different. Excuse me. Let's start up here on this strand up at the top. And in fact, let's write our strand orientation because this will become really relevant. Let's say that this strand is in the 3 to 5' orientation in the direction of the replication fork, which would make this complement, of course, the opposite, right? This complementary strand would be 5 to 3' orientation. That means if you were to look at each of these nucleotides, you would find that that 3' carbon is here and the 5' carbon is here, right, on that backbone. So that would make the new strand in the direction of the replication fork, which is over here, the new strand synthesized in the 5 to 3' orientation here in the direction of the replication fork. Now, importantly, let me just write down a couple notes here. An important thing to note is that DNA replication occurs in one direction only. Always the new strand is synthesized, is always synthesized from the 5 to the 3. So that 3' carbon is always the end of this newly synthesized strand because the polymerase works by adding on nucleotides to that existing 3' carbon. So it's always made in the 5 to 3' direction, period, end of story. This becomes a challenge because let's look at what's happening up here. For this one, if the complementary strand is, if the complementary strand to this one, or the newly synthesized strand, will be 5 to 3 in this direction, 5 to 3, that's actually no problem because the replication fork is over here to the right and this double-stranded DNA molecule is continuously opening in that direction. So you can essentially just have continuous synthesis or replication of that top strand. But note the bottom strand. The bottom strand's template orientation is 5 to 3 like this. That means in order for replication to go from 5 to 3 with the newly synthesized strand, that means that this newly synthesized strand is going to be replicated essentially backwards, right, in the opposite direction of that replication fork. So let's talk through how that process happens in just a second. Let's start up here at the top though. Once this double-stranded DNA molecule is open, then step number 4 is the addition of something called an RNA primer with an enzyme called RNA primase. So step number 4 is the RNA primase, again, an enzyme, lays down a short, this is usually about 2 to 3 or 3 to 5 nucleotides, RNA primer. Okay, so that's just a few nucleotides essentially to prime, just like you would prime a wall, right? You initiate this process by giving a little jump start. So here are a few nucleotides, 1, 2, 3, and it occurs in, or this primase and everything downstream will elongate or build this from the 5 to 3' orientation. So that essentially that 3' carbon is available, right? That 3' carbon is available for binding for that RNA polymer, or the key molecule in DNA replication, DNA polymerase to come and bind to. Sorry, that's a really bad picture. That's supposed to be an arrow. Let me draw my arrow again. Sorry, guys. Okay. Again, that 3' carbon is available right here. That's no better, 3' carbon. Okay, so this occurs at the start of replication, and now we can have the next player involved. Again, I'm just going to draw up here on this top strand. The key molecule involved in this whole process is called the DNA polymerase, and I'm going to draw it way down here. Key molecule involved in this whole DNA replication process, or step number 5, would be the DNA polymerase number 3. Number 3. And this job is to elongate the newly synthesized strand by adding nucleotides to the 3' carbon. That should say 3 prime carbon. So this guy will come and we'll add a nucleotide here to this 3' carbon, and then another one here, and another one here, and continue along this process, adding nucleotides that are complementary to the template, always to that 3' carbon, right? And it will continue indefinitely on this strand here as that replication fork continues to open all the way until the end of that replication cycle. At the very end of this process, so all the way at the very, very end, the last 2 key players involved in this process are the DNA polymerase 1, which will come and kick off all of those RNA molecules. What color am I on now? Let's make this in this color here. All right, so we have another player here that comes in downstream at the very end of this cycle. This would be step number 6. At the very end of replication, we have DNA polymerase number 1, and its job is to replaces that RNA with DNA. So now we have almost a completely synthesized strand, except there usually is a gap between this newly synthesized, what the RNA polymerase 1 brings in, and this existing elongated strand. So the last player involved in this process is something called ligase. Here's step number 7, is DNA ligase, and its job is to seal the gap. So it forms the very last bond between what was replaced and what is existing now to complete that double-stranded DNA molecule. So this is steps 1 through 7 in DNA replication. Now this is all fine and dandy for this strand at the top, as mentioned. We call this strand at the top, let me do a different color for you. We call this strand at the top the leading, it's not always at the top. This strand that allows continuous elongation is called the leading strand. The leading strand is where the orientation of the template is 3 to 5 in the direction of the fork. The leading strand template the leading strand is where the template DNA is in the 3 to 5 prime direction, or 3 to 5 prime orientation in the direction of the replication fork. And that means you can have continuous elongation of the newly synthesized strand in the direction of the replication fork because of this polymerase adding on to that 3' carbon continuously as this double-stranded DNA molecule elongates. Let's pause here for just a second. Let me show you a picture of that DNA polymerase, and hopefully this will make sense when you look at how it actually looks. So here is a picture of that DNA polymerase, the main player involved in this elongation process. Note that it sits upon a giant part of that double-stranded DNA molecule. And so its job is to essentially read the template nucleotides and allow only complementary nucleotides to come in. But it does so by bonding the incoming nucleotide to the existing 3' carbon. You can see it up here too, much better. If you recall the structure of DNA, again, it's important to remember how this guy looks. So in this one, you can see for the blue strand, this would be a 5' carbon. Here's 4', 3', 2', and 1'. And so this is 5 to 3 like this, right? The synthesis occurs, remember, from 5 to 3 for the newly synthesized strand, and it's because of exactly this. There's that 3' carbon. When it's not bound to anything, that 3' carbon has a hydroxy group on it. This allows for something called a phosphodiester bond to form between this carbon and this phosphate group by essentially donating these 2 phosphates to that hydroxy group. And yeah, there definitely is a video of this. So you can watch this stuff. I have some links on our Canvas page as well. And our textbook videos are on there too, so definitely go watch the stuff and you can see this process too. And again, so there's this incoming nucleotide and it bonds here at the existing 3' carbon, right? So this elongation continues in that direction in this picture by continuously adding on nucleotides to that 3' carbon. So this DNA polymerase is important for two things. One, to make sure that the correct complementary nucleotide comes in to bind to the template strand, and two, most importantly, to form this phosphodiester bond, or to form the bond between the incoming nucleotides and the existing nucleotides on that elongating strand. Okay, let's go back over to our whiteboard. It's always kind of shocking to see all this drawing. Okay, so this is all on the leading strand that I showed you, but I just mentioned both of these strands act as templates, but on the bottom strand in my picture, the orientation requires replication to go in the opposite direction of where that replication fork is. So we call this bottom strand the lagging strand, whereas the top strand is the leading strand. The bottom strand is called the where's my pen go? There it is. The bottom strand is called the lagging strand. Now, I keep using top and bottom, but that's only because that's the way I'm drawing it. In fact, in just a second, I'm going to have you guys break out into groups, and I'll challenge you to draw this backwards, where the replication fork is on the left. So pay attention here. This is all about strand orientation, right? You can draw this upwards, downwards, leftwards, rightwards, it doesn't matter. The biggest concept is to really understand strand orientation. The lagging strand, again, if the leading strand template is in 3 to 5 in the direction of the fork, the lagging strand template is 5 to 3. My doggy. 5 to 3, gotta go down. In the direction of the fork. And this requires replication to occur in the opposite direction of that opening replication fork. Okay, so let's draw this out. The process is going to be exactly the same. So we just saw again here, let's go back to number 1. Number 1 is topoisomerase, unwinding the DNA. Number 2 is helicase, unzipping the DNA. Number 3 is single-stranded binding proteins, prevents the reannealing. All that stuff is exactly the same. Now here is the next step, number 4. Over on the leading strand, we had this happening just one spot on the origin of replication to initiate this replication event. That's fine here, but on the lagging strand, the replication fork is continuously opening up that double-stranded DNA molecule. So it's kind of reverse time for a second, and let's go, let's say that this, right, this is the origin, this is the spot that opened up first. So as soon as there were enough nucleotides open, this replication event began. So step number 4, the addition of an RNA primer right here in that direction, right? Here's step number 4, is the RNA primer or primase. Primase lays down the RNA primer. Step number 5, just as we had up here, we have elongation occurring with the DNA polymerase 3, but remember we're going backwards here. So here we have DNA polymerase 3 adding on new nucleotides, right? From 5 to 3, so here's that 3' carbon, here's the 5' carbon here. So it's going in the opposite direction, and this will continue until it hits something, which let me explain to you up here. Over, as we are looking at this replication event, now notice that there are all these nucleotides that have been opened over here or exposed as that replication fork continues, but the replication event is over here. So this process continues over, or happens, repeats itself over and over and over again on this lagging strand as soon as new nucleotides become available. So here is the next sequence of nucleotides available, and we'll have this whole replication process again. So here again is the RNA primer, primase, laying down RNA nucleotides, right? Here again, we have DNA polymerase that binds to and elongates all the way down, right? DNA polymerase binds to and elongates all the way down. And as I mentioned, it'll elongate all the way until it's blocked. And now you can see how it's blocked. This nucleotide is the last one that can be added because this polymerase number 3, this polymerase can only do one thing. It can only add on nucleotides to that 3' carbon. That's all it does. So here is a 3' carbon. This DNA polymerase cannot bind this 3' carbon to the phosphate group on this. It can't do that, right? So it's stuck here. This is where this particular replication event will stop. And same thing here. And so as this replication fork continues to open in this direction, this whole reverse replication will just continue over and over and over again. And what you end up getting are these short fragments right here. So this whole fragment thing, it actually has a name. This is called an Okazaki fragment. So this is a complete replication event from start to finish. RNA primase, DNA polymerase 3, but we have these last 2 guys involved. Remember that we still have step number 6 and step number 7. So here we have, on both of these things, step number 6, the DNA polymerase number 1 will come here, right? 6 is DNA pol number 1. Same thing here. Step 6, we have DNA polymerase, pol 1, comes and replaces that RNA with DNA. And then we have our last step, which is that ligase, right? Here, I told you there is a gap here. So here is our last step, number 7, DNA ligase. So the ligase becomes super important on this lagging strand. You don't see it here, but it would happen again between these fragments that are generated. Ligase comes and essentially connects the Okazaki fragments on the lagging strand, right? So it becomes really important on this lagging strand to make a complete confluent double-stranded DNA molecule at the bottom. Okay, questions on what I just showed you here? I know that's a lot of messiness here, but hopefully you were following along. Anyone have a question you want to ask right now? All right, so what I'm going to challenge you to do, and again, good practice for that strange whiteboard thing, I'm going to separate you guys into smaller groups, and I want you to redraw this backwards. So I want you to put your replication fork on the left-hand side and just follow this through. Yeah, so again, I'm going to stick you guys into breakout rooms for about 10 minutes. And I want you to practice writing this whole replication fork backwards. So you're going to flip this thing around and you're going to put on the left-hand side is where that replication fork is going to be, and show me your leading and lagging strand and the steps 1 through 7 of how this process occurs. Oh, you want me to repeat the entire thing? That's funny. Is DNA polymerase 1 start on the RNA primase binding site? Essentially, DNA polymerase 1, yeah, it just comes in here. And RNA primase is only there at the very beginning to just lay down the RNA primer, and then it goes away. So DNA polymerase 1 comes back in right at the beginning, yeah, of where the RNA primer is, and just cuts out the RNA nucleotides and replaces them with DNA. So it does, yeah, start at the top of that. Hard to draw in a single linear thing, but yeah. At least label 1 through 7 somewhere, okay? So identify the key enzymes involved in that. Yeah. So work on this one with your partners, and I will tell you, if you do a good job, then you have already worked on your in-class discussion question that I'm having you do over the weekend. So make this your actual assignment if you want to, or screenshot it or something when you guys finish it, so you don't have to do extra work again. This is your in-class activity point assignment for that's on Canvas right now. Just a quick note that we noticed last week on Thursday in those breakout rooms, in order to get everyone to share, on that on the whiteboard someone who has, it looks like you have to have a computer, and if you click on share whiteboard and then at the top you'll have a control that says remote control. You have to click on that remote control button if you're the one that opened the whiteboard and just allow, there's something that says auto accept all requests or provide access control to other people or something like that. So just make sure once you get into the room, unmute yourself, someone assign themselves to be the host or something, just say, I'll do this, open up your whiteboard, share your whiteboard, and then click on that remote control to allow everyone to participate at the same time. And then everyone should have no problem going and adding to that thing. Okay, okay. Again, if you have a preference for breakout groups, let me go back to our, yep. If you have a preference, state it now. If there's any 1 or 2 people that you want to try to group yourself with, then as I split you guys up, I can rearrange you once you're there. But right now would be the time to let me know if there's anyone you want in your breakout group. Okay, I'm gonna put you guys 4 to 5 people per room. And I'll come visit you in just a few minutes as you work through this, and hopefully it won't take you more than 5 or 6 minutes to do this. Okay. Okay, we'll take that 30 minutes before this lecture discussion group starting Thursday, and also the 30 minutes after that. I'll give you guys like a 5-minute break or something to recoup, and then we'll just switch over and do completely lab discussion. And I'll be able to talk through the details behind some of the labs that we're going over, that we went over before, and the incoming labs. I think that will be much better for us. And again, I don't care which time you choose to participate or if you come to do both of them to get that lesson reiterated, but make sure that starting on Thursday that you plan that time within your schedule, either from 5 to 5:30 or from 7 to 7:30 to do a more intimate interface talking about the particular labs. And starting this Thursday, I will start that off by going over the genetic engineering one. Remember that that is due on Friday. So it would behoove you to definitely participate in that one. I'm not going to make any of these things mandatory, just so you know, but it definitely is in your interest to come and participate in that so that you can get a little bit more, one-on-one time. And in those shorter groups, I'll have you guys unmute yourself a little bit more so we can just have some discussions. All right, so since we only got through replication today, right now there is already posted a replication question, an in-class discussion question on Canvas. I think I have that one due on Friday. And then by Thursday, we'll go through the whole transcription translation. I'm sorry, on Thursday, I think I said that. We'll go through transcription translation. Please watch the video, the lecture video that's pre-recorded before then, so that when we get to this point, we can just go through the whole problem and do our shorthand, longhand discussion of all that stuff. And then I actually can get to mutations so that you have access to all of that discussion stuff on Thursday. I would appreciate that. Again, I will post lecture videos by the end of tomorrow. You'll have the rest of chapter 8 posted as the pre-recording lecture stuff, all covering gene exchange and mutations and repair machinery. So if you can get to that before Thursday, that's cool, but we won't cover that in detail until next week. I think that's everything. Please don't hesitate sending me emails asking me to change something, right? I'm trying to kind of do this all on the fly, so any input is completely appreciated. If you have any questions or comments and you want to stick around while people log out, that's totally cool. Laura, what I just mentioned, if you can watch replication, transcription, translation, those are the lecture videos that I posted that I recorded for you guys transcription, translation, and replication. If you can get through that stuff before Thursday, then on Thursday we'll essentially just be able to go through the discussion questions, which will take most of our time anyway. Genetic engineering, Pan, you're right. Genetic engineering is not going to be included on the exam. It's not in the workbook, or it's in there, but it's not part of the exam. So you definitely don't have to focus on that one. It's really more to assist your understanding of the genetic engineering lab. I used to have that as part of our lecture, but I don't anymore. So you don't have to worry about that part. Any other questions from you guys before we log off? Okay, if you want to hang out and ask me anything while people are leaving, you're welcome to. Otherwise, hopefully you guys stay safe, and I'll see you again on Thursday. Ciao.
Professor?
Yeah.
So what did you say we don't have to focus on?
Just the genetic engineering part of the textbook. So the textbook and the workbook has some of these questions too, but there's some parts about genetic engineering and the steps behind how you do that whole thing. You don't have to worry about that part for the exam, for the lecture exam. For the lab practical, you'll have questions of genetic engineering, but that's based on our lab and the P-glow plasmid and stuff that we'll go over on Thursday.
Great, thank you.
You're welcome.